How are complex neural pathways mapped with brain scans? Imagine trying to trace a tangled web of roads in a busy city without ever leaving your home. That's similar to how scientists map the brain's complex neural pathways using advanced scans. When we talk about brain mapping, the goal is to see how different parts of the brain connect and communicate. Conducting brain scans is the first step in this process. The most common method today is diffusion magnetic resonance imaging, or diffusion MRI. This technique tracks water molecules moving along nerve fibers, which helps us see the pathways they follow. Because water moves differently along nerve fibers, diffusion MRI can create a three-dimensional picture of these pathways inside the living brain. The data from diffusion MRI is processed with special models called fiber orientation distribution models. These models help resolve multiple fiber directions within a tiny brain area called a voxel. Think of a voxel as a small cube of brain tissue. Sometimes, fibers cross or intertwine within a voxel, making it tricky to tell which way they go. These models help clarify that. Then, tractography algorithms use this information to piece together the fiber bundles, revealing major pathways like the corticospinal tract, which controls movement, or the callosal projections that connect the two brain hemispheres. To make these maps even clearer, scientists often combine diffusion MRI with other techniques. One such method is polarized light imaging, or PLI, used on post-mortem brain tissue. PLI analyzes how polarized light passes through thin slices of brain tissue, giving a very detailed view of axon directions at microscopic levels. While PLI offers high detail, it's labor-intensive and doesn't show the full pathway origins and terminations in a living brain. Combining PLI with diffusion MRI helps resolve complex crossings and improves the accuracy of the maps. Another emerging technique is scattering light imaging, or SLI. SLI looks at how light scatters in thin slices of brain tissue to map nerve fibers with high resolution. It's faster and less costly than some other methods, making it useful for detailed studies without needing expensive equipment. Large projects like the Human Connectome Project use high-quality structural MRI combined with diffusion, imaging, and advanced processing to create detailed maps of brain connectivity across many individuals. These maps help both researchers and clinicians understand how different parts of the brain are wired. In practical neurotechnology, these mapping techniques are vital. They help identify disruptions in neural pathways caused by injuries or neurological diseases. They also guide brain enhancement efforts by showing the structural basis of brain functions. Techniques like diffusion MRI-based tractography, combined with PLI and SLI, give us a layered view of the brain's wiring at multiple scales. This helps scientists and doctors better understand how the brain works and how to treat or improve it. So, mapping complex neural pathways involves a mix of advanced imaging methods that visualize the brain's wiring in detail, supporting progress in neuroscience and neurotechnology.